Greetings to all the viewers. The subject what we chosen for the day is chemistry of hemoerythrin. It is the kind of bioinorganic chemistry. We are discussing about the pigment which carrying the oxygen. Oxygen transportation mechanism is performed by various metal bounded proteins, right? So we can say metalloproteins, right? These metalloproteins are accompanying that oxygen. Oxygen transportation in the living organisms are very essential, being oxygen required for all kind of metabolic activities and moreover digestion of the food and in order to transform food hidden energy into a kind of usable energy. This is the reason why that oxygen transportation is very essential. Some of the organisms, especially marine organisms, are accompanied with the pigment called hemoerythrin, where the coloration of the blood will be the purple. And that chemistry we are going to discuss in the session. Hemoerythrin simply abbreviated as HR, right? And the session is powered by AI, artificial intelligence powered session. It will be. Let me enter into the session of hemoerythrin. Intro regarding the hemoerythrin is provided over here. Hemoerythrin is the oxygen binding protein. It is the oxygen transporting device, we can say, and found in the blood of some marine invertebrates. Invertebrates denotes as the organisms which lack of backbone are said to be invertebrates, and they are laying in marine region, including certain types of worms and brachiopods, the kind of tiny organisms which ever not consisting the kind of bone system, skeleton system is absent over there. Unlike hemoglobin, which contains iron, here is hemoerythrin, which is also accompanied by the iron, but both the iron atoms are present within the molecule. Hemoerythrin contains iron atoms which are coordinated with the oxygen in such a manner that is forming a bridged one. Bridged oxygen is present, even deoxy hemoerythrin as well. Whenever oxygen bonded, that able to generate the peroxyl linkage. This is the way a kind of hemoerythrin is represented. You can observe the three-dimensional representation of protein and metallic part wherever deoxy form you can notice. This is the three-dimensional representation for hemoerythrin. And here is the manifestation. You can notice the iron atoms, iron 1 and iron 2 are located. Here is the oxygen which is binding in between the two irons as a bridged one in the deoxy form. And moreover, irons are combined with the kind of histidine, glutamate, and in either aspartame. These kind of amino acid moieties are surrounding the metallic iron, so that we can say these are metalloproteins. Inducing color change whenever binding with the oxygen, and they are especially meant for the oxygen transportation. Here is the oxygenated form. You can notice two iron metals are coordinated with the oxygen hydroxyl form, which is the bridging one. And in either oxygen, this is the oxygen what is supposed to be transported in the living organisms. This is the oxygen carrying capability. And here, these iron are bounded with the histidine nucleus. Histidine are accompanied by the imidazole moiety over there. Aspartame, glutamate, a kind of amino acids are also taking part of this kind of coordination with these metallic species. And they are forming a kind of web in between two metals. And there is a bridging oxygen and poly-coordinated systems around two iron atoms. So many amino acids are taking part of this one, like histidine, aspartame, and glutamate. Right. So this is the way a kind of hemoerythrin is represented. Let me go for the key features of hemoerythrin. What kind of features are accompanied by this hemoerythrin? Here you can notice deoxygenated form. Whenever oxygen is binding, that able to generate a kind of peroxyl linkage. This is said to be oxygenated form. And there is a reversibility. That's the reason why double-headed arrow indicated. The oxygenated form may convert it into deoxygenated. Deoxygenated will be transformed into a oxygenated, so that both are interconvertible in nature. Both the possibilities, both the directions, they are operative. Iron content. When you absorb the hemoerythrin, iron content is very essential. Deoxyform is accompanied by ferrous iron over there. Typically, two iron atoms are taken for one molecule and coordinated with the histidine nucleus. Nitrogens of these histidine taking part of this kind of coordinate covalent bond formation. In addition, aspartate and glutamate residues also forming a kind of web in between, so that it is a kind of hemoerythrin is a kind of complex molecule. It will be metalloprotein complex, we can simply designate. And the ability to bind with oxygen binding is reversible in nature. Both the forms are interconversible. Mechanism differs in the hemoglobin. The oxygenation in between is somewhat different. Oxygen, in case of that hemoglobin, directly pi bond will coordinate with this one. Dimolecular oxygen, whenever in contact with hemoglobin, what happens? Oxygen pi bond will coordinate with this iron, so that a kind of complex will be generated in hemoglobin is slightly distinctive in comparison to the hemoerythrin. Hemoerythrin, being two iron atoms, are responsible for this kind of oxygen transportation. That's the reason why whenever oxygen is binding with this hemoerythrin, that there is a provision to generate a kind of linkage among iron and oxygen that is giving rise to a peroxy linkage. And moreover, hydrogen bonding is also crucial in the oxygenated form of hemoerythrin. And if you observe the oxidation states of the iron before oxygenation and after the oxygenation process, here is a ferrous and later oxygenation giving rise to ferric. Oxidation state got increased and electrons removed from the metallic iron, giving rise to a higher oxidation, that is ferric form. So ferric form is oxygenated and peroxy linkage is very uh, hmm, common kind of thing in between this oxygen and iron metal together. This is the way hemoerythrin structural skeletal representation is provided. And if you concern with the color of the complex, being it is a kind of complex between the metal and the ligands of the various amino acids, obviously there will be a existence of color. Whenever deoxygenated form like ferrous, which is binding with the hydroxyl with the bridging bond type, and this kind of web is colorless in nature, where the deoxyhemoerythrin is the case. Whenever oxygen is supposed to be added, that giving rise to a kind of peroxy linkage and that intensifying the color into a purple one. Purple colored oxyhemoerythrin formation is possible. These are distinctive molecules by the oxygenation. There will be a change in its color. You can clearly observe colorless complex will be transformed into a violet pink colored complex because of the oxygenation process. In continuation to the intro regarding this hemoerythrin is used in the transportation of the oxygen. Oxygen transportation is the very important activity carried by this hemoerythrin, and marine invertebrates are accompanied with this kind of pigment for the oxygen shipping. It is an octamer. Octamer indicated for eight monomers are taking part of this one. Eight subunits are the part of this kind of hemoerythrin, and each subunit is accompanied by two individual iron in the form of ferrous. And the bridging oxygen is also the kind of molecule you can notice. Bridging hydroxyl will be there. Deoxyhemoerythrin contains two ferrous ions per subunit, and moreover it is the colorless one. Whenever oxyhemoerythrin taking oxygen giving rise to peroxy linkage, where the iron transformed into ferric one, bright reddish color, violet color is a kind of consequence because of oxygenation. These invertebrates also contains the monomeric form of the hemoerythrin located in tissues, analog myoglobin. Myoglobin is a kind of storing device, we can say. Just like myoglobin, they are also located in the tissues in the form of monomeric form. monomeric state. The binding of oxygen to the hemoerythrin and its release can be described by the following reaction. How the chemical representation, chemical equation is represented for the oxygenation of this hemoerythrin. That is accomplished. Each hemoerythrin is containing two ferrous ions. That's the reason why two ferrous are indicated. And oxygen will be added under acidified conditions so that ferrous will be transformed into ferric, being it is oxidation process. And another is supposed to be a kind of peroxy linkage. This is the way peroxy linkage is originated. Oxygen binding is accompanied by the transfer of two electrons. Each iron is releasing one electron individually and proton to this oxygen. Oxygen is accepting electrons from this iron so that ferrous to ferric transformation is noticed. This is the way chemical process accompanied by the oxygen transportation of hemoerythrin pigment. And marine invertebrates which are accompanied by the kind of this pinkish color plate are indicated over here. These many organisms which are said to be marine invertebrates. They are accompanied by the hemoerythrin oxygen transporting device. And here are the kind of specific distinctive representation is provided for these marine invertebrates. The species in specific are kind of Scipione worms. Scipionculid worms are mentioned over there. These are said to be a kind of oxygen transporting hemoerythrin containing species which are present in the marine region. And brachiopods also manifested, which are shell containing organisms, and some annelids. Annelids are said to be earthworm is the best example under that category. Earthworm, you can clearly notice that is said to be a kind of cultivating device for the farmers. So it is a very, very important composting agent. That is the natural composting agent. And here we are having some other species included with this Annelida phylum. Those are Elita succinia and Glycera species is located. And this is the kind of organism which everybody aware about. Hirudo medicinalis is the kind of strongly binding agent whenever it is in contact with the human skin. And Urachis Copo is another one, which is also the kind of marine invertebrate, the part of Annelida. And here, Hirobella species. So all these together comes under the annelidan species, where the backbone is absent, and they are the kind of worms, right? They are the kind of worms all together accompanied by hemoerythrin as a pink color pigment. Now let me introduce hemoerythrin activity, how it will work. It serves like a oxygen carrier. It's used to carry the oxygen, right? Oxygen carrier, and the storage of the protein is also the additional functionality by hemoerythrin, just like hemoglobin able to transport the oxygen and store the protein as well. And even myoglobin having a prime activity like the storage of the protein only. In vertebrates, how hemoglobin and myoglobin are working in the similar manner, this hemoerythrin in invertebrates that able to transport the oxygen and store the protein as well. This is the way hemoerythrin, the most important application will be the transportation of the oxygen. Oxygen transport and storage. Significance of hemoerythrin. Why we are supposed to study about this coloring pigment? What activity, what specificity it is having? Significance of this hemoerythrin to be discussed over here. Hemoerythrin functions as an oxygen carrier. It able to capture the oxygen from the environment and able to transport to the all kind of organs whichever present in those marine invertebrates. And even protein storage is also possible, just like hemoglobin and myoglobin. Even if the oxygen availability is extremely less oxygen available to environment, they are efficiently survived because of the hemoerythrin. Hemoerythrin is having that extent of flexibility. that able to manage the low levels of the oxygen as well, helping organism to survive hypotoxic condition. Hypotoxic condition stands for where the oxygen availability is extremely low. Lower concentration of oxygen is said to be hypoxic conditions of the oxygen level. There also the kind of hemoerythrin is effective to manage that low level of oxygen as well. This is the way oxygen transportation is the very essential activity done by this one, and moreover proteins to be stored by these hemoerythrin, they are able to store. They are the bank of the proteins, and moreover, oxygen levels are low. That condition also properly managed, efficiently managed by means of hemoerythrin. This is the way a kind of multiple activities taken by this hemoerythrin. Unique oxygen binding mechanism. Oxygen binding mechanism is very unique in nature. Unlike hemoglobin and myoglobin, where the linkage of the oxygen to the metal carried by means of coordinate covalent bond with the pi bond in between the two oxygens. Moreover, double bond may not be transformed over there. But in case of this hemoerythrin, the type of activity will be somewhat distinctive in nature. Hemoerythrin binds to the oxygen through non-heme diiron center. So here oxygen binding will be taken. One of the iron taking part of the bonding and another oxygen is also taking part of that. When this is the way a peroxy linkage is originated among the oxygen and iron, giving rise to a kind of cyclic structure. Being hydrogen bonding is also significant over this the binding of oxygen. Hydrogen bonding is also the kind of significant. In the deoxy form, hydrogen bonding is absent. In the oxygenated form, only hydrogen bonding is present. This is a kind of competitive exam oriented question. So how distinctive both the oxygenated and deoxygenated form. Deoxygenated form is the colorless one. Oxygenated form is the pinkish or violet color, and it is deoxygenated form is +2 oxidation state, and oxygenated form is +3. And here oxygen bridging in between two iron. Here also oxygen bridging. But moreover, peroxy linkage noticed in oxygenated but not in the deoxygenated form, and hydrogen bonding is present in the oxygenated form but not in deoxygenated. This is the way a structural distinctive factors are operated among the oxygen binding capability of this hemoerythrin. The binding mechanism involves the formation of peroxy bridge between the two iron atoms, making it biochemically unique in nature. Whatever molecule we generated, that is unique in the applicability of this oxygenated form of hemoerythrin in the biological field, biochemical field. Color change upon oxygenation. How the color change is noticed? Very simple. Colorless compound upon the oxygenation, giving rise to pinkish purple color. That is very distinctive, giving rise to a kind of oxygenated form. That simply visualized, so that need not to go for the identification where the oxygenation is carried and where the oxygen deoxy form is available. If the color is colorless, that simply is designated as the deoxygenated form. The purple color indicates it is the oxygenated form. And evaluationary significance is provided over here. The presence of hemoerythrin instead of hemoglobin in some invertebrate showcases changes in the oxygenation transportation mechanism in the animal kingdom. The transportation is obviously it is of distinctive in nature. Being invertebrates are the kind of tiny organism. They are surviving in the aquatic spaces where the saline salt content is extremely larger, so that they are having unique adaptations to their environmental conditions to survive. So that hemoerythrin is the kind of pigment which transport the oxygen in the different manner in comparison to the hemoglobin colorization. Highlights how different organisms have evolved distinct strategies to adopt their specific ecological niches. So whatever ecological species are kind of organisms which are surviving Being ecological sphere of the marine aquatic system is extremely large. It is the biggest ecosystem in comparison to the terrestrial one, so that multiple organisms are surviving together, though they are very tiny, not that extent developed. But the kind of species availability and the kind of survival are very larger in number and huge in the masses. That's the reason why there are adoptions towards the kind of survival in this kind of water environment, low penetration of sunlight, and the kind of oxygen availability all together effectively managed by the kind of species whichever available. Hemoerythrin is the kind of potential pigment which able to manage the kind of environment whatever present around these invertebrate species in this aquatic system. That's the reason why this is very evolutionary in nature for their oxygen transporting devices. And here you can notice the how oxygenation of these iron ferrous ions are taken and reversibly giving rise to a kind of peroxy linkage, and moreover cyclic structure being they are participating in the hydrogen bonding process, and these are said to be imidazole, which are extended with the histidine moieties, and these are said to be carboxylate, which are extended with the aspartame and glutamate kind of species. And this is the way hemoerythrin deoxygenated and oxygenated forms are represented. Biochemical and structural studies, how this is very significant in the field of bioinorganic chemistry. Being iron are the part of inorganic moieties only, so that the concept whatever chosen is the part of this bioinorganic chemistry as well. Due to its a unique iron coordination capability, oxygen binding mechanism, hemoerythrin has been studied extensively in the bioinorganic chemistry. This is not exactly similar with the hemoglobin and myoglobin. Rather, it is the kind of unique feature for the oxygen binding capability and the dimensions of arranging and its a way of working. That's the reason why it is extensively studied. Research is very in-depth research was carried upon these bioinorganic segment called hemoerythrin. Understanding the structure, function, provide the insights into the metal-centered oxygen transporting system. It is the kind of metal-centered oxygen transporting system and taken part of bioinorganic chemistry, so that hemoerythrin is very significant in the field of bioinorganic chemistry. Researchers are very thirsty about extending its applicability and mechanism of action as well. Biosynthesis. Now let me discuss regarding the biosynthesis of hemoerythrin, how it is fabricated in the living organisms of marine invertebrates, how it developed within the organism. So being it is the part of that living organism, biomolecule as well, so that hemoerythrin biosynthesis is taken for the consideration. Biosynthesis of hemoerythrin in that regard, the biosynthesis involves the assembly of the protein and the incorporation of the iron metal at their active site, giving rise to the final molecule called hemoerythrin. This is the protein what we are choosing. The protein is accompanied by histidine nucleus and aspartic acid nucleus and one more glutamic acid nucleus as well. They are supposed to be bind with the iron metal transition metal to be taken or to be assembled in such a manner metal centered protein will be developed, that giving rise to a kind of final product called hemoerythrin. This is the way biosynthetic scheme is provided roughly. Let me go with the individual steps which take it part of this hemoerythrin biosynthetic pathway. The step one, we have to proceed with the gene expression. So genetic matter matters for its a synthetic pathway, so that the genes encoding the hemoerythrin are transcribed into the mRNA and translated into the polypeptide chain. Transcript Transcription is meant for the combination of DNA with mRNA, so that giving rise to new strand of DNA. So that is the way transcription process will be accomplished, and the translation meant for converting DNA into RNA, and finally into the polypeptide chain. Finally, you're supposed to obtain the polypeptide chain by the process called translation, right? DNA is supposed to process with the transcription, giving rise to mRNA transcript. This mRNA transcript further follow with the translation process and giving rise to final polypeptide chain, which is said to be protein only. A primary sequence typically contains the histidine, aspartate residues that coordinated with the iron. The requisite amino acids, whatever polypeptide you are expecting for the formulation, so that is composed in terms of hemoerythrin, that is composed of aspartic acid and histidine together. They are taking part of this kind of transcription and translation process and giving rise to polypeptide chain. It is the kind of polymeric in nature. This is the way sequential arrangement of histidine and aspartate residues taking part, giving rise to polypeptide chain by means of gene expression process. Later, protein folding is a next step, right? Proteins are having multiple steps over there. Primary structure, secondary structure, tertiary, and even quaternary structure is present. Primary structure is related with what is the sequence of the amino acids in the protein fabrication. That is said to be a kind of primary. Secondary mean for whenever the molecules are taking curvature, there is a kind of curvature, there is a kind of folding we noticed over a secondary structure. Even the tertiary structure, the molecules are participating in the kind of hydrogen bonding, and even thiolinkages are taking part over there. So that helical structure adopted by the molecule, that is said to be a tertiary one. Even complicated arrangement comes under the quaternary. This is the way protein folding is a very important step in the hemoerythrin biosynthetic pathway, where translated strips are taken, translated polypeptide strips are taken. They are supposed to participate in the folding in order to achieve this tertiary structure. Tertiary structure meant for the kind of helicalness, a kind of complexity. Molecules are not laying in the straight chain manner, rather they are folding in particular dimensions. That is because of their hydrogen bonding formation, and even a kind of thiolinkages are also valuable. This is the reason why amino acids are whichever taken part of the polypeptide chains are twisted in such a manner, they folded in such a manner, giving rise to a tertiary structure to the protein. So that there is the certain cavities, certain hollow spaces are available, whichever suitable in order to accommodate iron metal in between. That is the reason why a complete folded molecule, a completely folded or twisted molecule is generated, that is composed by the polypeptide protein chain. In between, there are certain holes, certain cavities which are supposed to be accommodated by the metallic iron. This is the way protein folding is accomplished. Later, iron incorporation is the very important step. Whatever protein folding we achieved, we adopted, that is accompanied by the histidine, glutamate, aspartate as well. In between, there are certain cavities which are selective for the incorporation of metallic iron. Usually, iron taken in the form of ferrous form are transported into the cell through the metal ion transporters. Metal ion transporting devices are available within those organisms only. They're able to accompany these ferrous ions, and they able to transport to these protein cavities. These iron atoms are delivered to the apohemoerythrin, iron free. Apo means what? Where iron is absent. That is said to be apohemoerythrin. Through the metallocaparons. Metallocaparons are the kind of devices which able to carry these ferrous ions into this metal free protein skeletons. Iron binding proteins also playing over there, so that they are binding the iron with this kind of protein, so that iron incorporation segment will be accomplished. This is the way hemoerythrin manufacturing is possible, so that whenever biosynthesis of hemoerythrin is considered, metal center assembly will be fulfilled. Whereas metal taken over there into the entire tertiary structure of the polypeptide chain, so that metal coordinated assembly system will be developed. Metal center assembly, two iron atoms are incorporated into the polypeptide chain by means of coordinate covalent bond formation. And primarily, here the amino acids taken are the histidine and aspartate as well. And the iron atoms are the dinuclear, being two iron metals are taking part of one hemoerythrin, so that dinuclear system is the kind. A dinuclear center will be there linked by the bridging oxygen atom. Both are connected with the oxygen atom as well. In stock, it is provided with the metal centered hemoerythrin assembly is provided with it, which is the three-dimensional representation. And activation in the oxygen binding. Now, we synthesize the hemoerythrin. Now, it is very suitable for the binding susceptibility towards oxygen molecule. Whenever iron centers are properly assembled in between and the protein becomes the functional now, this protein is very active towards the capability to bind with oxygen, so that binding with oxygen will be the next step. Oxygen binding results in the color change from the colorless wherever deoxygenated form is there, and the violet pink color oxygenated form will acquire. As a peroxide bridge forms between the two iron atoms, whenever peroxide bond is located, that able to transform the complex color into the purple violet. This is the way color change indicated for the oxygenation process of hemoerythrin. This is the way biosynthesis is accomplished, and here you can notice the distinctive factors between the hemoglobin, hemocyanin, and hemoerythrin as well. Hemoglobin is a iron containing pigment which induces the blood red color, being it is the pigment inducing the color to that blood red color induced. Hemocyanin is the copper metal containing pigment. It will be upon the oxygenation, it able to transform into the sky blue color. Wherever hemoerythrin is the kind of pigment which able to change the color of the oxygenated form of that will be a purple violet. This is the way distinctive coloration you can notice for three different oxygen carriers, oxygen transporting devices, hemoglobin, hemocyanin, hemoerythrin, red, blue, and pink respectively. This is the way metalloproteins are represented. Now, let me enter into the kind of physical properties of hemoerythrin. Biosynthesis is successfully discussed. Let me bring physical properties of the hemoerythrin. Its a molecular weight is typically ranges between the 108 to 120 kilodaltons. Whenever it is acquiring octameric form. Octameric form indicates individual hemoerythrin 8 subunits to be binded, giving rise to 8 units containing octamer. Octamer is a kind of oligomer only, and it weighs in between the 108 to 120 kilodaltons of the weight. Monomeric subunits are ranging between 13 to 14 kilodaltons, only being it is octamer, so that weight is somewhat larger. And the coloration, deoxygenated obviously colorless, you already aware about, and oxygenated form is indicated with the purple violet color, that it is the kind of pink blooded animal. Solubility, highly soluble in the aqueous environment. Hemoerythrin, being it is the part of marine invertebrate, so that water solubility is obviously high, suitable for the oxygen transportation in the marine organism. Marine system is the larger, huge one. One third of the earth covered by the marine system only. That's the reason why the kind of hemoerythrin taking part of these marine invertebrates only. And iron content contains the two irons per one monomeric unit. and coordinated to the protein through the histidine, aspartate, and the glutamate residues. The histidine, aspartate, and the glutamate residues are taking part in between, and two iron metals will be there per unit. Here is the aspartic acid you can locate. It is the kind of acidic amino acid. Amino acid meant for amino functionality and carboxylic acid together existed. Whenever the molecule is having another additional carboxylic acid in that molecule, that is acidic one. Because acid base compatiated together, but one more additional carboxylic acid indicated for acidity for aspartic acid. Whenever you go through this glutamic acid, again you can notice there is a compensation of acid-base neutrality among the amine and carboxylic acid, but additional carboxylic acid inducing what the acidic nature once again, so that it is also the kind of acidic amino acid. Let me introduce the another amino acid taking part of this hemoerythrin will be a histidine nucleus, where you can locate amino acid, so that it is a neutral one, and it is accompanied by the heterocyclic ring system imidazole. This is the way histidine is a neutral one. These three are the essential components taken part of this hemoerythrin metalloprotein, and iron atoms are taken in the form of ferrous state. Deoxygenated form, whenever becomes oxygenated, that will be ferric. So that oxidation process is accomplished over there by the oxygenation process. Oxygen binding capability will be reversible in nature, deoxygenated to oxygenated, oxygenated to deoxygenated, both are vice versa. Both are possible reversibly. Forms a peroxy linkage whenever binding with the iron metal with the oxygen, and all are cooperative in nature, all are helping with each other, and they are existing in octameric form, so that it is the part of oligomeric in nature. Oligomer means for any polymer is composed by less than 20 subunits or 20 monomeric units. That is the part of that oligomer. Being it is octamer, you can designate as a oligomer only. Just like hemoglobin, here is also the kind of octameric nature is located, and all are cooperative in nature, all are helping with each other. And this is the way ferrous existing in Fe plus 2 form, and the oxygenated form will be oxygenated, giving rise to ferric. That is Fe plus 3 form upon oxygenation process. Now let me compare the hemoerythrin and hemoglobin together. Hemoerythrin is a marine pigment. Hemoglobin is a terrestrial pigment occurrence. Marine invertebrates like Scipion Culate worms and brachiopods are taking part of this hemoerythrin. Vertebrates, skeletal containing organisms are accompanied by this hemoglobin, where mammals and birds, reptiles are taking part of this one. We are also, humans also the accompanied by this hemoglobin because of our blood is red in color. Oxygen binding group in case of hemoerythrin will be non-heme diiron center will be there. Oxygen is binding with that one, so that non-heme diiron center is taking part of this hemoerythrin, whereas hemoglobin, heme moiety is directly binding to the oxygen. Protein structure is octameric in nature. 8 subunits are typically accomplished in a hemoerythrin coloring pigment. Whenever hemoglobin is considered tetrameric in nature, which is taken in the peripheral ring system, right? So, two alpha units, two beta units are there. Totally said to be 4 subunits are taken, which is tetrameric in nature. That is about the protein construction. And the iron coordination, two iron atoms are coordinated by the histidine and aspartate residues in case of hemoerythrin. Whereas iron atom bounded to the heme groups, and in case of hemoglobin, they bounded with these pyrrole linkages, which is said to be peripheral ring system. Color change. Colorless initially whenever deoxygenated form will be there, and changes to violet color upon oxygenation in case of hemoerythrin. Dark red color will be there in case of hemoglobin deoxygenated form. That will become a red color in hemoglobin. uh, Cooperativity. So that cooperative kind of linkage will be there in the hemoerythrin. High cooperative linkage you can notice in the hemoglobin. Oxygen binding mechanism is a peroxy bond formation to the iron is possible. In case of hemoerythrin, hemoglobin able to generate the direct bond with the oxygen to iron in the heme. This is the way hemoglobin, hemoerythrin are distinctive in nature. Molecular weight of hemoerythrin is 108 to 120 kilodaltons in the octameric form, whereas 64 kilodaltons weighed for the hemoglobin, being it is a tetrameric in nature. That's the reason why somewhat lower weight you can observe. Being larger octameric form weighs 108 to 120 kilodaltons for the hemoerythrin, 64 kilodaltons indicated for the hemoglobin. And the functions of the hemoerythrin oxygen transportation is very essential activity done. And storage in the low oxygen invertebrates also possible. Protein storage is also additional function that able to accompany. In case of hemoglobin, oxygen transportation in a blood and the tissues are also possible. They able to transport the blood. They able to transport the oxygen to the blood and tissues as well. Oxygen binding affinity will be moderate, not that larger in nature. Very strong bonds may not be generated in by means of hemoerythrin. Varies with the pH and ionic strength as well. If pH changes, binding modes also changes, and the ionic strength changes, their binding affinity also changes. In case of hemoglobin, oxygen binding ability is extremely larger, and pH matters for its binding ability, which is said to be a Bohr's effect, and even carbon dioxide level also matters for this one. Hemoglobin binding affinity towards the oxygen. Uh, in continuation to in continuation to that comparative study, now we entered into the segment of hemoerythrin compared with the hemocyanin. As of now, we compared hemoerythrin with hemoglobin. Let me compare hemoerythrin with hemocyanin now. Hemocyanin is also the invertebrate oxygen transporting device only, but some other organisms are taking part of this hemoerythrin and hemocyanin. Let me discuss the differences between these two. Occurrence of hemoerythrin are taken by the scipioniculate worms, and the brachiopods are the main organisms which are staying in the water, very saline water, that is marine invertebrates are taking part of this hemoerythrin. Whereas hemocyanin is biggest family in the universe, arthropods. Arthropods only the biggest insect family which is located highest population containing one. They are taking part of this hemocyanin. Crabs and lobster are the examples of hemocyanin, and even molluscs, octopus also accompanied by this hemocyanin. Oxygen binding affinity, non-heme diurein center taking part of hemoerythrin, whereas copper responsible for this oxygen carrying capability. Copper existing in cuprous in deoxygenated, cupric in oxygenated form. Protein structure will be octameric in hemoerythrin, as we already aware, being it is high molecular weight and heavier in nature. That's the reason why octameric forms are taking the part of that hemoerythrin. Multi-subunits, hexameric units, and even larger aggregates also possible. That depends upon the type of species. Mostly in general, they are hexameric in nature. 6 units are binding together. Metal ions or two irons are there in hemoerythrin, whereas two coppers are taking part of hemocyanin. This is the way distinctive factors are located in between hemoerythrin and hemocyanin. Let me continue color change. Hemoerythrin, we already know colorless will be the deoxygenated, violet pink will be the oxygenated. Whereas hemocyanin colorless will be deoxygenated, blue will be the oxygenated form. This is the distinctive factor again. Oxygen binding affinity, peroxy bond formation among the irons in case of this hemoerythrin. Copper is binding with oxygen. Copper binding with oxygen in case of this hemocyanin. Molecular weight is 108 to 120 kilodaltons, being it is octameric, heavier in nature. Very large molecular weight for hemocyanin. several million Daltons will be there. That depends upon the number of units, subunits are combining. Oxygen transportation and storage is the common function which is fulfilled by hemoerythrin, whereas oxygen transportation in the hemolymph meet for the resembling with blood. Blood is the name indicated for the vertebrates. Hemolymph is the fluid indicated for the invertebrates. Oxygen affinity will be moderate for hemoerythrin, moderate again for the hemocyanin as well. This is the way distinctive factors are mentioned for hemoerythrin, hemocyanin, hemoglobin, and hemoerythrin as well. Hope it will be very clear regarding the comparative study between the hemoglobin and the hemoerythrin, hemocyanin, and hemoerythrin as well. Let me introduce the applications of hemoerythrin, how it will be applied, how it will show its significance in the applicability of its functions in marine invertebrates, biomedical research and biotechnology. It's a precious chemical ingredient for the researchers in order to explore in the field of biomedical and biotechnology as well, right? Oxygen carrier, it will be blood substitute due to its oxygen binding capability. Hemoerythrin is explored as a potential model designing artificial oxygen carriers, right? This is the way oxygen will be binding with this purple color plate, and that oxygen able to form a bond in between that is incorporated, penetrated inside later. It will be transported to the requisite receptoric organs in the marine organisms, right? And unlike hemoglobin, it does not contain heme, reducing the risk of oxidative stress and toxicity. Toxicity may not be given by this kind of hemo. Erythrin, it is a very safe one. And hemoerythrin color change upon the oxygen binding, initially colorless, later it will be shifted to purple because of oxygenation process, and it is indicated with the double-headed arrow. Reversibility is the kind of criteria accompanied between the oxygenated and deoxygenated form. It can be incorporated in the sensor devices to monitor the oxygen availability in the biological and even in the industrial system, so that we can extend this pigment applications even in the field of biological systems where the oxygen availability is monitored. If it is becoming purple, so that you can assess it, there is oxygen levels will be higher. If it is staying in the colorless form, the oxygen levels are minimal. This is the way color change matter for its biological availability of the oxygen at the vicinity in the biological regions and industrial areas as well. This is the way oxygen monitoring sensor, it works like an oxygen monitoring sensor. Hemoerythrin will be. Let me go with the role model capability. Hemoerythrin serves as a model for the studying non-heme diiron proteins, how the binding capability will be there, and how distinctive, unique oxygen binding tendency will be there. It is very different when compared to the hemoglobin and hemocyanin as well. Metal homeostasis is a kind of thing, cellular management of the metal levels to maintain the bioavailability inside the cell, and minimizing damaging and cytotoxic effects, excessive metal buildup. Whenever metal inserted in between the proteins, that is called metal homeostasis, and it is said to be the level of metal, how extent it is required, that extent it will be managed well, is said to be metal homeostatic. That will be perfectly managed by this hemoerythrin and to be oxygenated, giving rise to a kind of unique binding capability. Hence, we can say it is a model for the metalloprotein studies. We can explore further. And biochemical and spectroscopic analysis will be accomplished, and how proteins are able to bind with two metallic iron in such a manner, and how coordination will be carried in between, and how this hemoerythrin entire coloring pigment will be generated in the living organisms. The entire studies, the entire analysis is very valuable in terms of biochemical and spectroscopic aspects as well. the interactive dimensions, interactive binding capabilities, and how they are stabilizing, what factors are driving in order to induce the stability. Entire study that in that purview also they are applicable. Insights gained can contribute to develop the synthetic metalloproteins as well by studying about their binding capabilities and stabilizing capabilities as well. It is possible to generate the mimicking pigments in the synthetic platform so that metalloproteins can be synthesized in the laboratory and they can applied in the various fields including with the catalyst. They works like a catalyst. They works like a oxygen carriers and they works like oxygen sensors as well. This is the way applicability of this same erythrin can be extended, right? Industrial and environmental applications are discussed over there. Its iron-based oxygenation mechanism inspires the design of biomimetic catalyst. Biomimetic in mean for whatever catalyst already present. The exact functioning catalyst will be developed in the laboratory. That is said to be what mimicking one, right? Exactly that is resembling, but not the same, though it is not same but can be developed in some other conditions, but resembling in its properties. The extent of similarities are extremely larger. That is said to be biomimicking catalyst. So whatever activity is provided by the hemoerythrin, same will be developed in the laboratory, and the kind of a catalytic activity will be resembling exactly. That is said to be biomimetic catalytic property, especially for the oxidation processes in the industrial purpose you can avail. It can be utilized in the field of grain chemistry for the sustainable catalytic processes, being it is the kind of biomolecule and having high affinity for the oxygen binding. That's the reason why it's effective utilization in the field of grain chemistry for the sustainable catalytic processes as well. This is the way hemoerythrin matters for the industrial and environmental applications as well. Environmental monitoring, being it is a bioindicator, oxygen levels will be monitored. Low level oxygen, high level oxygen indication can be better provided by hemoerythrin. It is the quantifying device in order to measure the levels of oxygen in the marine environments. This is the way environmental monitoring is also possible. Obviously, oxygen level in the water indicated for the level of pollution as well. This is the way the quality of the environment will be checked by means of this hemoerythrin, which is the coloring pigment which binds with oxygen reversibly indicates the color change. Color change matters for the oxygen concentration. Oxygen concentration means for the quality of the environment as well. This is the way it is the measure of the quality of the environment, biological indicator we can say. Pharmaceutical fields also taking part of this hemoerythrin. They are also taking the applications of hemoerythrin due to its iron coordination, oxygen handling properties. Researchers are very interested in order to investigate the potential to oxidate to reduce to mitigate the oxidative stress, right? Those who are suffering with oxidative stress, the kind of iron coordinating the metal will be inserted into that person so that they are able to relieve from the oxidative stress. This is the way it able to give the additional application in the field of pharmaceuticals and therapeutic potential as well, and can be studied therapeutic application. Hypoxic conditions are nothing but danger of the low level oxygens can be monitored and sensed by these pigments so that they able to give the indication alarm for the low levels of the oxygen. Hypoxic conditions will be quantified by hemoerythrin, and oxidative stress relieving capabilities then another advantageous application for this hemoerythrin as well. If you want to extend this applicability of hemoerythrin, evaluationary and comparative biology, understanding the oxygen transporting evaluation. So, oxygen transporting evaluation mean for Some organisms will be there, they simply transport the oxygen in general. If you're going with any other organism, then the extent of oxygen transportation is somewhat specific, somewhat distinctive, somewhat regulated manner. This is called what? Evaluation. If you go with any other organism further, again the oxygen transportation is still in a strategic route, that impressive in nature. This is the way from one generation to another generation, or else from one kind of species to another kind of species, there is a kind of variation in the terms of oxygen transportation. This is the way a kind of evaluation is accomplished by means of these hemoerythrin pigments in order to transport the oxygen. Just like we are valuing the various kind of comforts in the driver transportation, right? You may evaluate the water transportation, road transportation, air transportation as well. Different kind of transportations having their own comforts. In the same manner, different species are able to transport the oxygen in their circulatory systems. The kind of transportation is not exactly similar, rather it is the kind of evaluation from one organism to another, one generation to another, one species to another as well. This is the way a kind of evaluation we observed, we noticed in marine invertebrates, so that it is the kind of important observation regarding this utilization of hemoerythrin in the field of pharmacy, in the field of environment, in the field of industry as well. So, understanding the oxygen transportation is very essential. How it is binding, how it is transporting in the marine invertebrates. This is the way insights gained on the applied evaluation biology and study in the ancient marine invertebrates, how ancient and marine invertebrates transported their oxygen, and some species got modified, some species got updated in that how the oxygen transportation is effectively managed. This is the way we are going with the comparative study about the oxygen transporting evaluation. This is also taken part of this application of hemoerythrin. Let me give again the comparison between the deoxy and the oxy hemoerythrin. So there are certain changes, certain chemical physical property differences, and even a kind of color matter. There are so many kind of distinctive factors are operated between the deoxy and oxygenated form of the hemoerythrin. Let me bring all kind of comparative parameters indicated for deoxygenated and oxygenated form of the hemoerythrin. Oxygenation state, deoxygenated oxygenation form will be in case of deoxyhemoerythrin, there is no oxygen, so that we can say deoxygenated form, it will be no oxygen bonded to that hemoerythrin. In case of oxyhemoerythrin, oxygenated form, it will be oxygen bonded in the form of peroxy linkage. Deoxyhemoerythrin mean for the colorless complex, whereas oxyhemoerythrin is a violet pink colored complex. If you go with the iron oxidation state for the oxy form will be ferrous, that will be Fe plus 2. Whereas oxyhemoerythrin is accompanied by the ferric. Both the irons are transformed into ferric. Iron coordination. Iron atom coordinated with the histidine carboxylate residues as well. Histidine is the kind of amino acid. Aspartate and glutamate will be there. All together are coordinated for deoxy. In the same manner, this oxygenated form also having coordination with these histidine and aspartate and glutamate as well. In addition, we are having peroxide linkage between the iron metals. Peroxide linkage is generated by means of oxygen molecule. Magnetic behavior for the deoxygenated will be a paramagnetic, and paired electrons are located within ferrous transition metal ions, whereas ferric will be shifted to diamagnetic, all the electrons got paired up. Oxygen binding ability will be no oxygen found in the deoxygenated, whereas oxygen existing in a peroxy linkage form in oxygenated form. Spectral properties, no significant absorption noticed for deoxyhemoerythrin, whereas in case of oxygenated form, you can locate 330 nanometers and 500 nanometer peaks are observed for oxyhemoerythrin in your visible spectroscopy. It go with functions of hemoerythrin, deoxyhemoerythrin ready to bind with oxygen. Oxygen binding affinity will be larger, so that it tends to bind with oxygen. Whereas oxyhemoerythrin is ready for the transportation. It is able to transport the oxygen and moreover able to store the oxygen as well. It will store the oxygen as well. Structural change. Open relaxed conformation is given for deoxygenated. Slight conformational variation noticed for the oxygen bounded hemoerythrin. Slight changes are observed. Stability will be less for the deoxygenated. Highly stable oxygenated form will be. This is the way distinctive factors are given for deoxygenated and oxygenated form of the hemoerythrin. By this, we successfully accomplished the entire session. This is regarding the hemoerythrin, that is the metalloprotein, bioinorganic chemistry it will be. And I hope you enjoyed the session. I must thank you, all the participants who are very curious about the content. The session conducted by Dr. Kavita Kottireddy Kotta. I'm academician and researcher in the entire content and series of sessions, whatever conducted earlier are available in the channel World of Competitive Chemistry. Hopefully, it is very helpful for the scientists who are working in the specific domain what we are discussing and researchers who are in the field of bioinorganic chemistry, organic chemistry, and industrial chemistry, and biotechnology as well. This kind of session will be very informative, pharmaceutical field as well, right? And the aspirants for the competitive exams and any entrance examinations as well, this will give the most informative inputs regarding the kind of requisite points regarding this chemistry. So everybody enjoyed the session. Thank you very much for your patient listening.